And today is all anatomy, right? So our next lab quiz on Tuesday is kind of like the respiratory system. It'll be just structures. And then the second quiz will add the histology, all right? So I'm not showing any of that as part of lecture today, but you're welcome to do it in lab or do it in lab on Tuesday next week. So we're just going to look at the structure of the kidney, the outline of the blood supply to the kidney, and then the functional unit of the kidney. Right? On Tuesday, we're going to come back to those same structures and go over them again as far as how they work. So we're going to talk about what the glomerulus is and the proximal convoluted tubule. And then on Tuesday, we'll look at it again as to what's the metabolic activity that's occurring in those structures. All right, so today is pure anatomy. And, yay. And in fact, sometimes when I give the respiratory test as a separate test, I'll combine those two lectures and we'll do structure and function at the same time. You guys are get a little extra time because we'll go over the structures again as we do the function. So urinary system, as you recall, we've already looked at the embryology of it just a bit because we've talked about these structures in the development of the gonads. So remember the pronephros and mesonephros and metanephros terms? You should at least be familiar and say, yeah, I've seen that before. And just real briefly, the important material that I want you to remember from this, and by important material, that reflects, because remember it's a take-home, a lot of this detail you can look up, but we still have the comprehensive, okay? So that's what I still want you to remember for that aspect of it. The pronephros is, of course, the first kidney structure to show up. It's shown up here, closer to the head, but it's actually longer than that. So it actually runs the length of the embryo. And it is I'm going to put non-functional, but in parentheses, because it does have function, but it doesn't function as a kidney. All right? In other words, there is no filtration. There's no exchange between blood, small particles moving out of the blood and moving into the kidney tubule. So its major function is induction. It induces or influences the tissues around it to develop into the next kidney structure, and also induces the development of the gonads, the ovaries and intestines. Okay, so that's its major function. Influences the further development. So we do see ducts. We do see an association. So here in the pronephros, we do see these outgrowths of what are going to form our functional kidney structures, but there's no filtration that occurs. There's no separation by particle component. Okay? And as the pronephros is degenerating, we see the development of the mesonephros. There's no other organ in the body that has three separate different structures. The brain develops as a brain. We don't have one brain and then it disappears and we get another brain. Okay? The same with the heart. We don't have one heart and then that disappears and another heart forms. So this is the only one that I'm aware of. We have the mesonephros, which develops as a separate structure, and this is the first time we actually see filtration, a functional association between blood vessels and the nephron tubule. And of course, the mesonephric duct remains in the male to form our efferent ductules and vas deferens and those components of the male reproductive pathway. And then that disappears. Both of these, so as this is disappearing, and it starts to disappear first in the head, all right, and as that's disappearing, we see then the development of the mesonephros, and then finally the true kidney, the one that remains, the metanephros, also has, of course, a functioning filtration. But interestingly enough, that doesn't develop in this caudal-cephalic relationship, but actually grows up out of the definitive bladder. All right, so this is what's going to become our functional filtration structure. So if we look at, again, this diagram, remember in that clay animation that we saw for male and female reproductive structures? So here's the mesonephros, and then starting growing out from the bladder, here's the true kidney, and then it grows up towards the abdominal wall. So the pronephros and mesonephros start high in the abdomen and disappear. That's why we have our gonads starting up high. The ovarian or testicular arteries actually start up high on the abdominal aorta and go all the way down to the pelvis or the scrotum. But the actual true kidney starts down here and migrates upward. Totally weird components for all of that. Okay? And that essentially is what I want you to remember from the embryonic development. We have these three separate stages. The first one, even though it might have an association between the blood supply, doesn't have this cup development appearing. There's no actual filtration occurring between the embryonic kidney structure and the true kidney. All right, so on the first page of lecture notes, the third page of your packet, I have listed the functions of the kidney. And one of the study guide objectives or the study review material asks you to list what part of the kidney, what part of the nephron works with each of these areas. Most of us are familiar with the waste excretion. Okay? The kidney is one of the urinary system, is one of our several systems that has an excretion function. What would be some of the others that function in excretory aspects? Getting rid of stuff.
Glands.
Glands where?
Skin.
Good. So the skin, we have actually even urea is a component of sweat. All right. What else? Pardon?
Just tears.
Tears certainly have chemicals in them, but they're not considered to be part of the waste aspect as much, just because there's not very much of it. Okay. What else is a major excretory?
Digestive.
Digestive. I was going to say poop in case you forgot. Okay. And then we just finished one. Pardon? No? Carbon dioxide in the respiratory system. Carbon dioxide is a waste product of carbohydrate breakdown. Okay? So those are urinary, the integumentary for skin, the digestive system, and the respiratory system are ways in which we get rid of waste byproducts from the body's function. So for the waste excretion in the kidneys, it is primarily nitrogen byproducts. So which of our foodstuffs contain nitrogen? Carbohydrates, do they contain nitrogen? It's a gas, but proteins, all right? Carbohydrates are just carbon, hydrogen, and oxygen. Fats are carbon, hydrogen, and oxygen. Proteins are the only ones that have nitrogen. Okay, they usually also have sulfide and other components. But we see these byproducts as urea, uric acid. That's the most concentrated form. Ammonia. And creatine. And so these can be toxic to the body as they build up. And use the water of the urine to keep them diluted as we excrete them. All right, regulation of the ionic composition. So ion or electrolyte regulation. What are the first ions that you think of when you think of kidney function?
Sodium.
Hydrogen?
Hydrogen.
Really? Okay, that is true. Well, we'll talk about that under pH. What else?
Sodium.
Sodium. And sodium has an intimate relationship with potassium. So when the kidneys are getting rid of sodium, they keep potassium. And when they're getting rid of potassium, they retain sodium. All right? So we won't discuss them specifically. One of the reasons I do a take-home on the urinary, so I can ask questions about diuretics. But there's a class of diuretics known as potassium-sparing diuretics. Okay? So additionally, those are probably the primary ones because we use this to regulate water movement. But we also, the kidneys are an important site for calcium regulation and calcium absorption. And to a lesser degree, magnesium. Okay. Regulation of blood depth.
Why did they say that coconut water is good for?
What, coconut water?
Yeah.
Because it has the closest, the number of the content is closest to what your biological, physiological values already are. Even Gatorade is hypotonic. So drinking a ton of Gatorade can actually result in water poisoning and making your tissues too dilute as far as the ions are considered. Now, blood glucose, probably I should take off the word regulation because that implies neurological neurotransmitters or hormones that can change how much glucose is retained or excreted. And what we really have is if you have normal amounts of glucose in your body, all of that will be removed from the urine and returned after filtration. If you have excess amounts, then you start ending up with glucose in the urine, okay? But it doesn't function, in other words, diabetics can't get rid of excess glucose by just peeing it out. So it's not regulation so much as we see, you know, glucose being returned to the body or not returned to the body, but not under a regulatory fashion. And as Corey said, pH. So remember we were talking about acidosis on Tuesday, and I said if you had pneumonia and you retain carbon dioxide, you're going to end up with too many extra hydrogen ions in your blood as additional carbonic acid is formed. And because the problem is respiratory, you can't breathe faster to get rid. The whole reason you have respiratory acidosis is because it's a respiratory problem. And so your body will respond to those additional hydrogen ions by adding them to the filtrate. So getting rid of them in the urine. Okay? Urine pH can range from about 5 to 8. So it can become much more acidic than the blood. If you have alkalosis, which means your pH is too high, then your body stops adding hydrogens and can actually add back some of the bicarbonate ions, okay, or get rid of. So this is, again, is done not by hormones, but it's driven by levels. So there's a high number of hydrogens, more hydrogen gets added to the filtrate. If there's not enough, then less of it gets added. Blood pressure. We've already talked about this? Yes?
It's actually going back to the other functions.
Uh-huh.
So for the blood glucose, it
In other words,
returns
additional if the glucose values are normal, glucose is a small enough particle that it will pass out of the blood into the filtrate, what we call urine, before we're done modifying it. If those values are normal, the body will take back all of that glucose from the filtrate. So it's abnormal to have glucose in your urine. If your levels of glucose are high, because your body's not able to have the cells take it in, then that excess glucose is not returned and stays in the filtrate. Not enough to keep your blood values normal, so it doesn't cure it, it doesn't balance it out that way like it can help to balance pH. But you do see that effect. It will get rid of some of that excess glucose.
Okay, and also the whole electrolyte thing, basically we have if we think about it, like calcium is more of a, is it like a mineral or something?
What?
Or is it like more of a mineral or?
Yeah, but it's an ion, it has an electron. Sodium is a mineral too.
Okay.
But
okay,
what I say is the kidney actually forms calcitriol. It takes vitamin D.
Okay.
It's one of the sites for forming calcitriol, which is a hormone that regulates calcium levels. And so somebody who has kidney problems needs to be careful about the amount of calcium in their diet that they have. So sometimes a kidney or renal diet will limit the milk products that they can take because they have lost that ability to do so. All right, well, we've looked at the heart and cardiac output. We talked a little bit about the kidney's role in regulating blood pressure. We talked about 3 hormones that affect kidney function. And you guys remember what they are? Renin is not a hormone, but aldosterone. All right, so aldosterone affects the kidney to increase sodium reabsorption. And that functions to elevate blood pressure. What was another hormone? ADH? ADH increases water reabsorption directly without sodium, and that also increases blood pressure. And then we had another hormone that lowered blood pressure. Anybody remember what that one was? As soon as I tell you the organ that makes it, the heart. Atrial, natriuretic, exactly, natriuretic peptide or protein. So A and P, atrial. Remember, this is our word for sodium in Latin. Peptide or protein. And that decreases sodium reabsorption and decreases blood pressure. All right? Now, this is not an altruistic activity of the kidney. It's not doing it just to help the rest of the body. But blood pressure is very important to the function of the kidney. Why? Filtration. All right? Filtration uses force to separate particles by size. If blood pressure is too low, you have low urine output. You don't have enough force pushing those particles across the filtration membrane of the kidney. We'll talk about that more on Tuesday. If blood pressure is too high, you can actually damage the cells that form the filtration membrane. And people that have high blood pressure in pregnancy will have proteins showing up in your urine, which shouldn't be there. Proteins are large and they're too big, they shouldn't be crossing the filtration membrane. But that high blood pressure can open up the spaces and allow large molecules through. All right, so the kidneys are intimately involved with regulating that blood pressure because it contributes or deletes from their function. However, this last function, I have not figured out why the kidneys are so interested in erythropoietin. EPO? why the kidneys over any other organ are interested in the oxygen carrying capacity of the blood. So if you live in a higher altitude, Mexico City, down the Andes, you know, a base camp in Everest, this starts to take effect after about 5 days at high altitude. You'll start to see an increase in erythropoietin, and you'll form additional red blood cells. Remember, we talked about if you have you're at high altitude with low atmospheric pressure of oxygen, you can only bind, you can't completely saturate every hemoglobin molecule. So you make additional hemoglobins so that each one is not saturated, but because you have additional ones, you can still carry the same amount of overall oxygen. Okay? Yes?
Could it be related to an additional regulation of pH via PCO2?
It could be pH, it even could be blood pressure, because with polycythemia, you'd have additional pressure force. But to me, it seems more like the brain would be more interested in, to use an anthropomorphic term, or even the heart muscle itself, on how much oxygen is available.
Yeah.
I haven't, and I need to do a little bit more research. I don't know the cells that form this, how they detect oxygen levels. Is it the peripheral chemoreceptors in the aorta that also then signal the kidneys to increase? Just haven't found a really good connection. You can come up with, ideas, but I haven't found a good physiological connection between those two. So it's always been kind of a mystery to me. But this is what we'll be looking at as we go through the structures of the kidney next week and talking again about them and their function. So I'll kind of tie back that. Here's the structure that's doing this. Here's the structure that's doing that. All right, so you can look at its overall component. All right, so the word peritoneal here refers to what membrane? What's the whole word? What's the membrane that lines the abdominal cavity? Parietal peritoneum. Okay? So on the lungs and the heart, we had pleura for the heart, for the lungs, pericardium for the heart. So in the abdominal cavity, we have peritoneum, right? And we first used that term as the outer covering of the uterus, which we call the periometrium, which is attached to the top of the uterus. So here, and we'll do this more thoroughly when we get to the digestive system. So here where it's attached to the body wall, it's the parietal peritoneum. But our abdominal organs grow into that and become surrounded by the visceral peritoneum. However, some of our organs do not grow out into it. They stay attached to the abdominal wall, and therefore they're covered only on their anterior surface by peritoneum. So the pancreas, parts of the large intestine. And when that occurs, they are identified as retroperitoneum. Okay? So they have only a portion of that organ associated with the peritoneum. Which brings us to, in its anatomical position, if you were to have kidney surgery, what would be the best approach, surgical approach, to get to the kidneys? Would you make an abdominal incision, come in through the abdominal cavity, through the back? That's a lot of important structural muscle. It's actually a lateral. So someone who's had kidney surgery or a kidney transplant, the incision is lateral, just under the ribs. Okay. The position of the kidneys, if we look at them on the posterior abdominal walls, is the top portion rests against the lowest two ribs, so ribs 11 and 12, your floating ribs. So sometimes individuals will have those two pairs of ribs removed so they can have a smaller hourglass figure. I think that surgeons that would do that are quacks because that's the only bony protection that the kidneys have, right, are those two ribs. Other than that, it's all muscular, and the two major muscles are the quadratus lumborum. I don't know if you covered those in Bio 430, but they go from the iliac crest to those lower ribs. So they would go from about here to here. Quadratus refers to the shape of the muscle, okay, kind of quadrangle. And then the other muscle is this psoas major. The psoas major crosses into the hip, helps you flex your thigh. Quadratus lumborum maintains the stability, so when you walk, your hips are stay balanced and don't rise and fall up and down. Okay? So those are the muscular supports to the organs. Now, someone that has a kidney transplant, normally the kidney is not placed back into its original position. Okay? It has as support in that position what's called a perirenal capsule. It's just adipose tissue, but it's not a strong support. So individuals who do a lot of horseback riding or drive a truck for a living, tractor trailer rig, especially here in California with our awful roads, all that bouncing around can actually cause one or both kidneys to start sliding down the back of the posterior abdominal wall, because it's not a really strong support, and that can cause a kinking in the tube, the ureter, that the urine is transported down to the bladder. So that becomes even more likely if another kidney is put in there and it doesn't have its natural support. And so most commonly that kidney the new kidney would be placed here along the iliac fossa with the shortened ureter. So it's not as slight. It's going to slide down there anyway, we might as well just put it there. Okay? All right, so now let's look at the general pattern of organization of the kidney. I don't know which came first, the kidney or the kidney bean, but it looks very similar to a lymph node. Obviously the size is not, but it has a cortex and a medulla and a hilum. Okay, so those same words that we've seen previously. Let's kind of draw this organizational pattern here. And I'm going to make this a little bit bigger so I have room. Now, there's no dotted line in the real kidney, okay? I'm just putting it in here to help us. So this region here is known as the cortex, all right? There is a thin capsule of dense irregular connective tissue on the outside that it's difficult to peel off. And then we have a series of, they look like triangles here, but they're actually pyramids, three-dimensionally. Okay, it's just a cut in a cross section. And some of our faculty do kidney dissections. I haven't in probably 12 years because they really don't show a whole lot that our plasticized kidneys don't show. Plus it stinks of urine, ammonia. And I don't know how people can eat kidneys when, nobody would drink urine, but they'll eat kidneys. I would say nobody. If you look online, there are people that will do a health practice of, not colonic cleansing, that's the wrong word, but they'll drink urine. Urine is actually sterile. It comes from your blood. It doesn't pick up bacteria until it's exiting through the external opening of the urethra, unless you have a kidney function. Okay, but in your bladder, it really should be sterile. And so this outer portion here is the cortex, and then these white structures here are the renal pyramids, or medullary pyramids. They have a striated appearance because the structures that form them are parallel structures. And you'll see that by the time we're done with lecture today. So, these medullary pyramids make up the medulla. So the medulla would be essentially the area that contains these pyramids. So one of these structures is known as a medullary pyramid, completely different from the medullary pyramid of the brainstem, obviously, in the medulla oblongata. The tips of each of these have openings from the tubes that are carrying the urine, the filtrate out, and so that tip is known as, if we looked at it on end, it would have openings from these tubes carrying the filtered blood product, and then that blood would, that filtrate would drip out. But this tip of the pyramid is known as the papilla. Okay. So each papilla then drains into a channel. Let me draw that in a different color so let's visualize it a little better. And if you're familiar with urinary epithelium, transitional epithelium, that's what we find lining these. Just kind of do it this way. So each channel that each pyramid drains into, say one of these right here, is known as a calyx, and it's identified as a minor calyx. Right? And then two or more minor calyces join together to form a major calyx. Right? So here would be two minor calyces, and this would be their major calyx. Here would be three minor calyces and their major calyx. Right? Once the filtrate, the fluid that has been filtered from the blood, exits the tips, the papillae of the pyramids, and enters into the minor calyx, that's the first time we actually call this fluid urine. Okay? From the process time that it's filtered from the blood until it actually enters into the minor calyx, it's just called filtrate, because we're still changing its contents, either the concentration or the actual content. So we're taking the glucose out, or we're adding additional hydrogen ions, so we're modifying it, we're taking water out. So we don't call it urine until it actually drips into the minor calyx. So if I were to draw that over here, so this would be a minor calyx. And this would be urine. Up here, it would be filtrate. Okay? And then all the major calyces gather together into this trumpet-like region, which is known as the pelvis or renal pelvis. Okay? And then that exits through the hilum of the kidney, narrows, and passes down towards the bladder. And this is the ureter. So everything that I've drawn in blue is going to be lined by our urinary epithelium, which is transitional epithelium. Okay, all the way down to the bladder through the urethra. So if we look at a model, okay, so here would be cortex. This would be a renal or medullary pyramid. Here would be a minor calyx. What you're looking at right here is the papilla of a pyramid that's behind the screen coming out at you. All right? And then here would be a major calyx and the renal pelvis and the ureter. Okay? So that's kind of the major structure. Now, there's another term that I will identify, define for you, that we really don't use, and that's the sinus. All right, so the sinus is this cavity right here that supports the calyces and the pelvis. And I kind of thought the best way to explain that is if you think of these calyces and pelvis as a swimming pool. Let's say you're going to put a swimming pool in your yard, and it's not the free form, you're going to buy a fiberglass swimming pool. Okay? My sister lives in Tennessee, and she decided to put this, for some reason she had to order it from Australia. I don't know why she thought that was a better fiberglass pool, but you dig a hole, okay? That hole would be the sinus. The pool that you set down in that hole would be the pelvis, right? And then you backfill that in, and that backfill is adipose tissue. All right. So clinically, if you ever get that far along in specializing in urinary diseases, there are two different types of kidney infections. So one of the sinus and the other of these structures. Okay? But you probably won't have to worry about it before then, and I really don't ask lab questions about it. It's just people see the word sinus and get confused. What's the difference between the two? And that's what the difference is. Okay? So it's that non-functional kidney tissue that supports the structures of the minor calyx and major calyx. All right?
What did you say all the blue was again?
The blue is transitional epithelium. The lining of the calyces and the pelvis and the pelvis looks a little anemic.
So it's not just the urinary bladder?
No. So that's what you learned it in 430. Most people in 430 don't talk about the calyces. I don't. But it's the ureter, the urethra, which is the tube that carries the bladder carries the urine from the bladder to the external. So remember the penis, we have the spongy urethra, the membranous urethra. That's transitional epithelium. The bladder's transitional. The ureters are transitional, the pelvis, and the calyces. Because remember, part of the purpose of having the transitional epithelium is because it's stratified, it's going to be resistant to reabsorption. We've now concentrated the urine. We don't want to reabsorb that urea back into the body. The fact that it can stretch is most important in the bladder. Okay? But that multiple layering is useful as soon as we have a urine formed and it's highly concentrated with urea, uric acid, ammonia. We don't want to just reabsorb that back into the body. And that stratified layers in the transitional is protective for that. All right, now let's look at the blood supply, because it's a little hard to filter. We don't have the blood coming in to be filtered, right? So one of the largest branches off of the aorta, in fact the largest branch off of the abdominal aorta, is the renal artery. It's about the size of my thumb, because the entire volume of blood passes into the kidney multiple times a day. So we have a large volume going in, and so let's bring that in here. It's coming off the aorta, and it usually has a couple of branches, 2 or 3 branches before it gets into the kidney. And then it branches a few more times. You don't have to worry about the segmental arteries, okay? And then we have, from the segmentals, we have branches going up. Usually a single branch will pass between each pyramid. So where these vessels are found between each pyramid, they are known as interlobar. Okay? So this would be an interlobar artery. The segmentals would be down here, the ones that we're not going to worry about. So where I would ask you to tag them if we were to look back at that model again, these would be the interlobar right here. Okay? Right here and right here. This area is identified as renal columns, and I don't talk about that. We don't have to worry about renal columns. But that's where we would find the interlobar arteries. Then each interlobar artery is connected where the medulla, whoops, wrong color, where the medulla and the cortex change. So at the base of each pyramid, the interlobar arteries are connected. And because this forms an arch, it's identified as the arcuate artery, which you should forget because it's ARC. Okay? From each arcuate artery, so again on our model, that would be right here, right here, right here, forms an arc. Okay? So arcuate artery. From each arcuate artery, we have straight vessels that extend at right angles out into the cortex. So these are known as interlobular. A lobule is smaller than a lobe. So we have interlobar, and then we have interlobular. Some textbooks are starting to call them cortical radiate, which means they're radiating out into the cortex. So if that's easier for you to remember, that's fine. But it's not where I expect you to know that the cortical radiates are also the interlobular. I'll just accept either or. All right, now, I'm going to take this section right here and draw it larger, okay? So this would be the arcuate still. Here would be the cortical radiate, or interlobular. So all of this would be cortex. And this would be medulla. All right? Everybody with me? I'm just taking that smaller section and drawing that larger portion. So coming from the interlobular artery, we have a series of branches that have lost most of their smooth muscle. We only have 1 to 2 layers left. So these are known as afferent arterioles. Then we lose all of the smooth muscle. What vessel comes after an arteriole? Capillary. So these capillaries are formed as tufts, balls that are coiled and twisted back and forth on each other. So this is known as a glomerulus or glomerular capillaries. Then, leaving the tuft of capillaries is another arteriole. Since it's leaving, it's called an efferent. Now, the efferent arteriole can do one of two things. If it's up high, in other words, farther away from the medulla, the efferent arteriole will lose its smooth muscle and form a large bed of capillaries known as peritubular capillaries. If it's close to the arcuate artery, instead of forming peritubular capillaries, it will dip down into the medulla and form a loop back up again. Where it goes into the arcuate vein. And this structure is known as the vasa, which means vessel, recta, which means straight. Those are found only in the medulla. And the peritubular capillaries and everything that I've drawn up there are found only in the cortex. So that when we look at histology on Tuesday, that will help you figure out where you are. Now the peritubular capillaries will go into interlocular veins, which will drain back into arcuate veins and just follow them back out. The vasorecta will just go directly into the arcuate veins. Now, when you're ready, I want to erase the kidney and those vessels that I've drawn already and redraw that same diagram on the far right and add my nephron, my actual kidney, functional kidney unit to it in a diagram that looks going to look like this. Okay? So again, on Tuesday, we're going to come back and talk about these same structures again and what they do. So we'll look at the kind of epithelium that forms the kidney structures and what they do and how they do it. Okay. So can I erase the kidney, the big kidney?